In this video, we will discuss the three ways by which lipid structure and distribution relates to membrane function. And it will be membrane curvature, linked to the shape of lipids, the asymmetric distribution of lipids across the membrane, and the fluidity of membrane that is determined by the lipid composition. Let's start with lipid curvature. Recall from the first video that in a curved portion of a membrane, if the outer leaflet is occupied predominantly by lipids that have a cylindrical shape, then in the inner leaflet of the membrane, you need to insert lipids that have a conical shape in order to maintain the integrity of membranes. The implication is that the distribution of lipids across the inner and outer leaflet of a membrane will be asymmetric. For example, we can expect that phosphatidylcholine, which have a cylindrical shape, will be more abundant in the outer leaflet, while phosphoethanolamine, which have a conical shape, are more abundant in the inner leaflet. We can confirm this hypothesis by determining the lipid composition of the inner and outer leaflet of a membrane, for example, of the membrane of a red blood cell. We can see that indeed there is more phosphatidylcholine in the outer leaflet and more phosphoethanolamine in the inner leaflet. We can also, from this analysis of the lipid composition, see that sphingomyelin is also enriched in the outer leaflet of the membrane. And phosphatidylinositol and phosphatidylserine, which are lipids with a net negative charge, are enriched in the inner leaflet of the membrane of red blood cells. Now, in red blood cells, the asymmetric distribution of lipids is a critical factor explaining the interesting biconcave shape of red blood cell that is so important to the function of red blood cells. We can also see that a change in the distribution of lipids has important physiological consequences. For example, we can see in an aged red blood cell that the asymmetry is broken down. Phosphatidylserine appear at the surface of red blood cells. They become now predominant in the outer leaflet. This is a signal to macrophage to clear out these aged red blood cells.
So how is the asymmetry of the lipid bilayer maintained?
We know that lipids are mobile in the plane of the membrane. They can diffuse laterally very easily. However, the spontaneous flip of lipids from one leaflet to the other leaflet is unfavorable because the hydrophilic head of a phospholipid would have to cross the hydrophobic core of the lipid membrane. In fact, we have an active movement of lipids from one leaflet to another leaflet, which is driven by proteins. And there are three different types of proteins. There is a type of protein called flippase, and they move lipids against their concentration gradient from the outer to the inner leaflet. Then we have floppase, which also move lipids against their concentration gradient, but this time from the inner to the outer leaflet. And finally, the third type of protein is called a scramblase, and it moves lipid in both directions, always following their concentration gradient. So in a way, flippase and floppase maintain the lipid asymmetry, while scramblase will erase lipid asymmetry. Now we can establish a relation between the composition of membranes and the site of lipid synthesis. The bulk of lipid is synthesized in the endoplasmic reticulum and in the Golgi apparatus. And lipids follow the same pathway as secreted proteins. So some are made in the endoplasmic reticulum, then they travel to the Golgi apparatus via vesicles, then they go across the Golgi apparatus, and then will travel from the Golgi apparatus to the plasma membrane, once again using transport vesicles. The only difference is that in this case the lipids will be embedded in the membranes of the ER, of the Golgi, and of the transport vesicles. Now we can assume that because these membranes have a common origin, that their composition will be very similar. And in fact, when we look at bar graphs that show the distribution or the composition in lipids of these different membranes, at a first glance, they are very similar. But if we, in fact, take a closer look, we can, for example, see that sphingomyelins are more abundant in the Golgi membrane and in the plasma membrane than in the endoplasmic reticulum membrane. And that makes sense because sphingomyelins are synthesized in the Golgi system. What is also interesting with sphingomyelin is that their synthesis occur on the luminal phase of the Golgi stacks. Therefore, they will be integrated into the inner leaflet of the Golgi. And when they travel to the plasma membrane in the vesicles, they will be also in the inner leaflet of the membrane of the vesicles. But as the vesicle fuses with the membrane, the inner leaflet of the membrane from the vesicle becomes the outer leaflet of the membrane of the cell. And that explains why sphingomyelin is more abundant in the outer leaflet of a membrane than in the inner leaflet. So in a way, the asymmetric distribution of sphingomyelin is explained by its mode of synthesis. So the lipid composition affects membrane fluidity. The simple rules that we discussed when we talk about the melting temperature of fatty acid apply to phospholipids. In a membrane, if you have phospholipids with saturated long fatty acid tails, these phospholipids will form dense, regular, packed structures. This membrane is then in what is called a paracrystalline state with low fluidity. So in contrast, if you have a membrane that contains phospholipids with short, unsaturated fatty acid tails, the packing is looser, the membrane will be more fluid, and the membrane is said to be in a fluid-like state. At physiological temperature, a membrane exists in a state that is intermediate between the paracrystalline and the fluid-like state. Another difference in the lipid composition of membrane is the abundance of cholesterol in cell membrane compared to the membranes of the Golgi apparatus and the endoplasmic reticulum. Remember that cholesterol is this odd cousin in the lipid family. It's a structural lipid, but it has a structure completely different from other structural lipids, with this very flat structure made of multiple ring structures. The reason why cell membranes are enriched in cholesterol is the transformation that happens in the vesicle that transport lipids from the Golgi apparatus to the membrane. During their travel to the plasma membrane, a fraction of phosphatidylcholine is replaced by sphingolipids and by cholesterol, explaining the abundance of cholesterol in cell membrane. Now, cholesterol molecules affect the fluidity of membranes. Remember that cholesterol is a very flat structure that can intercalate between the fatty acid tail in the membrane. Now, the rigid structure of the cholesterol molecule will alter the mobility of fatty acid that have short, unsaturated fatty acid tails. It will decrease the fluidity of this membrane. In contrast, cholesterol will also interfere with long saturated phospholipids, and it will disturb the tight packing of these lipids and will prevent the transition to a paracrystalline state. So it will increase the fluidity of the membrane. Cholesterol keeps the membranes in the sweet spot. It keeps them fluid, but it also maintains their rigidity. In conclusion, the composition of lipids and their distribution are important contributing factors to the structure and the biological function of membranes.